Konnichiwa guys, welcome to our channel Loops of Biology. I'm Yogiraj Jakkal, taking you all to the next one-shot video on cell cycle and cell division. So today we are going to summarize the complete chapter of cell cycle and cell division, which is one of the important chapter as per the NEET part is going to be considered. So in this chapter, we will be covering the topics such as introduction, cell cycle, mitosis, cell death, and finally the meiosis will be covered along with all the NEET questions the previous year and some of the questions which are out of the NEET previous year's syllabus. Alright. So let's start with the introduction. Now, as we can see here that there are two cells which are separating from each other so that the new cells are going to form. So the cell division is necessary. It is necessary for formation of new cells. For formation of new cells. As it might look interesting to you that during this particular phase, we have known the cell as the cytoplasm unit along with a cell membrane, and there is a presence of nucleus inside it. So there is a cytoplasm, nucleus, and a cell membrane. So whenever the cell is going to divide, this particular elements are going to get split. So these are basically split into two units. However, if we are dividing this particular cytoplasm continuously, then that particular content will get over at the time. So it's not happening whenever the cell division is taking place. Now, how is it possible? The cytoplasm content, is not reducing. How? By duplicating this particular content. So the cells duplicate this content. All right. So let's move to the next part, that is the cell is unit of life. We have seen that along with its detailed analysis and its need to undergo division for new cells to produce. Obviously, whenever the reproduction, which is a fundamental unit of life, is basically seen, so in that particular case, during reproduction, the cell has to divide so that new cells are going to form. Now, every new cell is coming from a pre-existing cell. So that concept is called as omnis cellulae cellule. That is, new cell arises from pre-existing cell. So that is nothing but a concept of cellulae cellular. Now, along with that, the zygote undergoes division to form multicellular organism. If we are trying to see how multicellular organism work, so in multicellular organism, there has to be formation of gametes. That is nothing but male gamete and female gamete. And after fusion of this gamete, there is going to the formation of zygote. And this zygote undergoes cell division to form the organism again. Alright, so new organism is basically formed. So the system is well organized with a proper signaling pattern. Now, what does this mean? That whenever a cell is ready for the cell division, that particular cell will send a signal. That cell will send a particular signal within itself so that the particular cell division process will start. So in this case, the system is well organized. That means the neighboring condition or the surrounding condition, is determined by cell, and then cell division take place. Alright, got my point? Okay. Cell division is essential for cell to divide and genetic variation to occur. So there are multiple type of cell division which are taking place, such as mitosis, mitosis and meiosis. So if we are talking about cell to divide, then this is common for all cells, for all cell division type, all cell division type. However, if we are talking about genetic variation, this is seen only through meiosis. And can anyone guess that which type of cell inside a body will divide? Is there any specific type, or all the cells of body will start dividing? So which cells divide in body of an organism? Body of an organism. So there is only a single type of cell which is going to divide inside a body, and that cell is called as stem cell, which has property of totipotency, that is tendency to divide and differentiate into a specialized cell. Alright, so let's move to your next part, that is your question. The bacterium divides every 35 minutes. So if we are considering a bacteria, that is nothing but your prokaryotic cell. So this is prokaryotic cell, and it is unicellular in nature. It is unicellular in nature. So it undergoes cell division in such a way that one cell divides to form two cells. So unicellular organism undergoes cell division to give rise to two cells, and these two cells are formed after every 35 minutes. So there is, after every 35 minutes, there is a formation of two cells. Alright, so there are right now, initially the cells are 10 raised to 5. Initial number of the cells is 10 raised to 5 cells per ml. Alright, is grown for 175 minutes. So the total time is nothing but 175 minutes. So can you guess how many generation of the organism will form? Can you guess how many generation, the number of generations of organism is going to take place? That is 175 divided by 35 minutes, which comes to be 5. Alright, so there are total 5 generation running in the particular content. Alright, so in this case, the total number of organism, that is nothing but bacteria, has one simple formula, that is 2 raised to n. So there are total 10 raised to 5 cell, that's why multiply by 10 raised to 5. So there is 2 raised to 5 multiply by 10 raised to 5, that is nothing but 32 multiply by 10 raised to 5 cells are going to be formed. So that is option number 3. Alright, so let's move on to the next part of the cell division. That is nothing but your cell cycle. What do you mean by cell cycle? Is nothing but it is a sequential events taking place in the life of cell. Alright, any cell which is undergoing cell division, it has to undergo through some of the sequential events, and this sequence cannot be disturbed if the cell is undergoing the process of cell division. So this sequential event occurs in the cell division process. And in this particular case, if the cell has to divide, it has to generate some of the proteins which are required. The proteins are basically coming from the RNA, and there should be a doubling of DNA content. Now, why the DNA content has to get doubled? Alright, so this content is double so that equal distribution of DNA content should take place. DNA content should take place. So there is, after the division of cell, whenever I am forming two cells from a single cell, then whatever the DNA content was there in a single cell has to divide equally within the two different cells, and there should not be any partiality within the particular cell for the DNA content. All right. So there should be organelle has to duplicate. Similarly, as the DNA content is double, the organelle number are also going to get double, and cell might take rest when there is no need of cell division. All right. This rest is very, very important. If suppose any cell, the cell that cannot undergo cell division, then where should that particular cell should go? That will be the question, and that is called as G0 phase. And G0 phase is also called as resting phase, and some literate can say it as quiescent phase. Alright, so there are total three names where whenever the cell is not undergoing cell division, that is G0 phase, resting phase, or quiescent phase. If you are talking about all the events occurring inside a cell cycle, out of which first event is called as interphase. It is called as interphase. So there is an event called as interphase, and that interphase is comprised of G1, S, G2 and G0. Alright, so these components are coming under your interphase. Now what are these particular phases? If you are talking about G1 phase, that is called as first gap phase. So this is called as first gap phase. Now what is going to happen inside the G1 phase? Let's see. If we are running the particular cell division, we have seen that we are going to require proteins. Alright, and these proteins are required in the S phase. Alright, so whatever the protein are required for S phase are formed within your G1 phase. Alright, so there has to be formation of RNA, proteins. Then this, after the proteins are formed, that means the cell is going to grow. Alright, so there is growth of the cell, and as we have seen, the organelles are going to get double. So organelles are double. Alright, simple. This is nothing but your G1 phase. All the cells after cell division are entering within the G1 phase itself. Alright, so G1 phase contains all the cells after cell division is completed. All the cells are entering within your G1 phase. Alright, then after moving to your S phase, the S phase is very, very important, as this particular phase contains DNA replication. So in this process, DNA replication is going to take place. So in this S phase, it is basically the DNA is synthesized and replicated for its copy, so that the DNA content will get double. So in S phase, the copy of DNA is synthesized. S for S phase, S for synthesize, you can remember. It is synthesized as the DNA content, gets double. Alright. Then moving on to the G2 phase, whenever we are considering G2 phase, it is your second gap phase. It is your second gap phase. Now this gap phase is correlated with your M phase. It is correlated with your M phase. Whatever the proteins are required for M phase, whatever the proteins which are required for M phase, those proteins are basically synthesized inside G2 phase. So in G2 phase, there is going to be your protein synthesis, and this synthesis is comprised of cyclins and CDKs. Alright, these two proteins are important for the M phase, cyclins and CDKs. Now what is the full form of CDK? That is nothing but cyclin dependent kinases. Cyclin dependent kinase. I hope you are remembering the kinase name. If not, go and check enzyme part in one short video for biomolecule on loops of biology itself. So this CDKs are nothing but cyclin dependent kinases. These kinases are very important for addition of phosphate group on substrate. Alright. Then moving to your G0 phase, as we have seen, this G0 phase is called as a resting phase, or it is called as quiescent phase. It is called as quiescent phase. In this phase, whatever cells are entering, all are non-dividing cells. The cells do not require division. Those cells are basically entering inside G0 phase, and final phase is nothing but M phase. This M phase is divided into two categories. This is called as period of division. This is nothing but dividing phase, right? And this dividing phase is comprised of karyokinesis, that is division of nucleus, and there is cytokinesis, that means division of cytoplasm. So there is going to be, during the M phase, there is division of nucleus and division of cytoplasm is going to take place. Now this M can be mitotic or it can be meiotic, dependent on the type of cell. If we are considering a total cycle of G1, S, G2 and M phases of interphase for 24-hour cell division. So in this particular case, the G1 is going to take a time of around 8 hours. Around 8 hours. If you are talking about S phase, it will take a time of 6 to 8 hours. Then there is a G2 phase which will take time of 2 to 5 hours. 2 to 5 hours. And there is finally M phase which is going to be completed into 1 to 3 hours. So the fastest phase to complete in a complete cell division process is nothing but your M phase. Alright. It will take very less time for the particular process to complete. Alright. There are some checkpoint questions which we will be covering in the question part. So the DNA replication in bacteria occurs. As we have seen, the DNA replication is nothing but doubling DNA content. It is nothing but doubling of DNA content, and this is occurring within S phase of interphase. Alright, so this answer should be option number 1 during S phase. Next, when the cell has stalled DNA replication fork, that means the replication has been started slightly before. Alright, so the DNA replication fork is basically formed. Which checkpoint should be predominantly activated? Now, whenever we are dividing this particular DNA, and we are duplicating or making a copy of it. So replication fork is important for formation of copy of a DNA. Copy of a DNA. Now, if I am making a copy of DNA, I should be very much aware, and the cell should be very much aware, that this DNA should not have any damage, right? So during this formation of a copy of DNA, the DNA should not have prior damage. Otherwise, the duplicated DNA will have the same damage continuing, and those will be transmitted in the next cells. Alright, getting my point? So in this particular case, the DNA is checked. The DNA is checked for its damages and mutation before entering S phase. This is very important, guys. This is very, very important. Before entering inside S phase. So which phases are correlated? That is, first of all, G1 was there, then S, then G2, and finally there is going to be M phase. So before entering inside S phase, that means there has to be a checkpoint at this particular point, and this is comprised of G1/S. So the answer will be option number 1. That is, G1/S checkpoint is activated so that it can check for its DNA damages and mutation before entering inside the S phase. Next question. A somatic cell has just completed the S phase of its cell cycle as compared to gametes of the same species has. A somatic cell that has just completed the S phase of its cell cycle as compared to gamete of the same species has. Now, if I am talking about gamete, alright, whenever I am talking about gamete, generally they are haploid in nature. Now, what do you mean by haploid? That means there is only a single set of chromosome with only 2 arms of chromatid. 2 arms of chromatids. So that is basically called as chromatin fiber. So that is nothing but called as chromatin fiber. So whenever I'm talking about S phase, so after completion of S phase, we have came to know that DNA content is 7. So the amount of DNA, is double. So wherever the double or the twice of the amount of DNA, those will be my option. So twice amount, 4 times amount, this will be eliminated out. So option number 3 is completely wrong. Now, what about chromosomes? That is, in S phase, the chromatid are double to form a sister chromatid, on same chromosome. That means number of chromosomes are basically same. So the chromosome number remains same and DNA content will be twice. So it's the same number of chromosomes but twice amount of DNA. So option number 2 is my correct option for this question. Let's move to the next part. during which phase of cell cycle the amount of DNA in the cell remains 4C. The amount of DNA will remain 4C if the initial amount was denoted as 2C. Alright. So that is very interesting question. If I'm talking about S phase, alright. So there is G1 phase, then there is going to be a S phase, then there is going to be a G2 phase, and there is going to be a M phase. These are 4 different phases. If you are talking that initial amount is 2C, initial amount was 2C for DNA content. Now this will be doubled inside S phase, so it will become 4C. Alright. This 4C will continue inside your G2 phase, and M phase is basically formation of 2 cells. So this 2 cell will contain 2C amount. Alright. So there is basically division of this 4C content inside the M phase. But as we are talking about the complete phases, so after completion of M phase, there will be 2C content inside the M phase. But till the particular nucleus is divide. Till some point, the M phase also has 4C content, but as the question is saying during which phase of cell cycle the amount of DNA in the cell remain at 4C. So there should not be any remaining of 4C at the end of M phase. That's why M phase is deleted. S phase is still the point, but S phase is combined with G1. So this option will be gone, and there will be only G2 phase will be my correct option for this question. So let's move to your first type of cell division called as a mitosis. If we distribute this particular part as A, B, C, D, and alright, we are just labeling the images. So a mitosis is basically very primitive type of cell division. So this is your primitive type of cell division. Now, what do you mean by primitive type? That is nothing but in this particular cell division, you are not going to see any fancy stunt happening. Alright. So there is just an elongation of nucleus. After elongation, as you are stretching the rubber, at one point the rubber will break. Alright. That point is called as break point. The point where nucleus is stretched. There will be a break point. And at that particular point, your nucleus is broken down. Nucleus is separated. Alright, in 2 nuclei. Whenever I am going to get the 2 nuclei, then there is going to be a division of cytoplasm. So then there is division of cytoplasm, and finally you are going to get 2 cells. This is the basic process of a mitosis. The main thing is that the nucleus is stretched. That means there is elongation of nucleus. Has to occur. Alright guys. So let's see what is a mitosis saying. At point A, the nucleus is prominent with a proper shape. As we can see, there is a nucleus in orange color which is in a proper shape right now. There is a spherical shaped nucleus. But when we are moving to the B part, the nucleus become elongated and stretched. So there is elongation process start. Process of elongation is basically start. At the C point, whenever we are looking at the diagram C, that means in this particular phase, as B here, the nucleus are connected together, and in C there is proper nuclear separation is seen. That means the nucleus is break into two parts. So there is formation of two nuclei. So there is formation of two nuclei. And finally the cytoplasm is divided, but that division is completely on the luck. It can happen or it can divide in any plane. In any plane. This is one of the important part. This is not regulated. And then I am going to get two daughter cells. Now, as we are talking about mitosis, it is a primitive type of cell division. That means there is no regulation. No regulation on cell division, for equal distribution of cellular content. Equal distribution of cellular content. This is one of the important things if we are talking about mitosis. Alright. Let's move. What are the significance of mitosis? This particular process of cell division will only occur in unicellular organism as it is a primitive cells. Then there is formation of clones. Now what do you mean by clones? That means they are genetically and phenotypically, that means based on external appearance, same as parents. Alright, so there is a formation of clones. Total time for the process is very less, as this is very quick process. There is a quick process. Now why it is quick? Because there is interphase is not occurring. Then you are going to see that doubling time for cell is very less. Alright, so doubling time is very less. Then this particular type of cell division can be seen inside a fetal membrane cell and abnormal cells. So these are basically one of the examples for your mitosis process along with your case of amoeba. Alright, so let's move on to your next one of the important type of cell division called as mitosis. Now in mitosis I am going to get two identical cells. There is no as such a problem that the cells are not identical to each other. Alright, so they are exactly identical. So they are exactly identical even in the basis of cytoplasmic content. Even on the basis of cytoplasmic content. Alright. Now the chromosome numbers are maintained. Now what does that mean? That the total number of chromosomes present inside parent cells, the same number of chromosomes are present inside the daughter cell itself. So there is number of chromosomes are same in parent and daughter cells. Alright. So whenever we are keeping the particular number same, such type of cell division is called as equational division. Alright. So in this case here, chromosome number are maintained. That's why it is called as here equational division. The total process is divided into karyokinesis and cytokinesis, as we have talked about it. That is, karyokinesis is division of nucleus, and cytokinesis is nothing but division of cytoplasm. So first of all, there will be a division of nucleus taking place, and then further the cytoplasm is going to get divided. Now the cells with any ploidy can enter into this particular cell division. Now what does that mean? If I am talking about the ploidy is nothing but total sets of chromosomes present in a cell. If suppose the set is only 1, this condition is called as haploid. If the set is 2, then there is a condition called as diploid. And if set is 3 or more, 3n, 4n, 5n, then there is going to be a case of polyploid. Alright, so these are one of the ploidy conditions of any cell. So these cells are basically with any ploidy can enter into the particular process called as mitosis. Alright, so from mitosis, after a cell division, one cell give rise to 2 cells. This is important as per the calculation part is concerned. If you get any question regarding calculation based on mitosis, then you should be knowing that one cell is going to give me 2 different cells. Alright. So what would be number of chromosomes of alluron cell? Now what do you mean by alluron cell? So alluron is a covering of protein around endosperm. The food which is useful for a developing zygote, to convert it into an embryo and finally into an organism. So that is called as endosperm, and this endosperm is triploid in nature. So similarly, the alluron layer is also triploid in nature. Alright, from this particular information, we came to know that alluron cells are basically triploid in nature. Now there are 42 chromosomes in its root tip cell. Now the root cells are basically diploid in nature, and they are having 42 chromosomes in total. So if we are talking about alluron, which is 3n, you can divide it as it is as 2n plus n, or you can just multiply the n with 3. It depends on you. So if I am keeping 2n as it is, that is 42, plus half of this particular chromosome as n, that is 21. So there will be total 63 chromosomes are present inside the case of alluron cells. So this is option number. Let's move to the karyokinesis process of mitosis. If you are talking about any karyokinesis, that is nothing but division of nucleus. So in this case, in this particular case, there are total 4 different phases. And these phases are mentioned as below. That is prophase, metaphase, anaphase and telophase in the sequential order. So this order for your karyokinesis phases is sequential. There is no sequence breaking if we are talking about any of this particular phase. First of all, the prophase has to take place, then metaphase, then anaphase, and finally the telophase is going to occur. So let's see sequence wise what are these phases. If we are talking about prophase, the prophase is the first mitotic phase taking place in any dividing cell. So this is your first mitotic phase. Now, in case of first mitotic phase, the cell has to enter from interphase. The cell has to enter from interphase. Now, as we know, if suppose one cell with n number of ploidy in G1 phase is entering, while moving to G2, it has to undergo S phase, where your DNA content is going to get double. This content is maintained as it is, and finally this is distributed inside two cell as NN. So the parent was with the same number of chromosomes, and even the daughter cells are with the same number of chromosomes occurring. So now this prophase is basically after the interphase. So already the DNA content has been doubled. So cell copies its DNA content. Then in that particular phase, your prophase is divided as two different phases, that is early prophase and late prophase. If you are talking about early prophase, that means just in the beginning of prophase, the centriole and centrosome doubles. Now, what do you mean by centriole? You must follow my channel and go to the particular part of cytoskeleton, where you will be understanding what is centriole and centrosome. Alright, please check out that particular video on our channel. So centriole is present inside the centrosome. The centriole is present inside centrosome, and it is important for releasing of microtubules. So microtubules are released, and these microtubules are nothing but, guys, these microtubules are nothing but your spindle fibers. These are nothing but your spindle fibers, which are very, very important if I am talking about a cell division. You will be understanding that in the metaphase of this particular cell division process. Then your chromatin fibers. These chromatin fibers are nothing but a thread-like structure of chromosome. So there is one more word, that is nothing but your chromatin fibers, which are nothing but your thread-like structure, of chromosomes. Now this will start undergoing coiling and condensation. That means the total height of the chromatin fiber will get shortened, and the coiling will start to take place, so that the thickness of chromatin fiber will increase. So in this case, the coiling is basically increasing the thickness. So in this case, the thickness is increased, while the condensation is shortening the height of chromosome. Alright. Now, in the late phase, after the centriole is getting double and your chromatin fiber undergo coiling and condensation, your nuclear membrane and nucleolus starts to disintegrate. So there is breaking down of nuclear membrane and nucleolus. Now, why is that happening? Because the nucleus has its own cover, and this centriole and centrosome are present outside nucleus. So if we have to separate the particular chromosomes, then we are required spindle fiber to attach on the chromosome, and this spindle fiber are released by the centriole, which are present outside the nucleus. Alright, so as soon as we are breaking the nuclear membrane, we are exposing all the chromosome to your spindle fiber, so that the particular pulling force will be applied and the chromosomes will get separated. Alright, so we have to expose. So this nuclear membrane is disrupted because chromosome has to exposed to the spindle fiber, fibers, so that those chromosomes will get separated properly, and then they will be distributed in the next nucleus, which is formed in a particular new cells. All right. So your question, guys, what happens to the disintegrated part of nuclear membrane and nucleolus, and why centriole is duplicated? Now we have to think out of the box for this particular chapter. If you are talking about breaking down of nuclear membrane, that means there are the pieces of nuclear membrane present within your cytoplasm. Will they remain same, guys? Will they present inside the cytoplasm, or will they get absorbed inside the cytoplasm? Then which particular organelle is going to play a role? Are you going, or are you thinking about anything? No. So if you're talking about the parts of nuclear membrane and nucleolus, we know that this part are going to contain your proteins and lipids. Now there was only a single cell organelle which was handling both of this. That is nothing but your endoplasmic reticulum. So these parts are basically absorbed inside endoplasmic reticulum. So in the beginning, we don't want this part to create any hindrance in the process. That's why this particular parts are absorbed inside endoplasmic reticulum and kept safe. As soon as the process is over, whenever we have to bring and develop a new nuclear membrane, then this part will be again released from endoplasmic reticulum itself. All right. So this is absorbed inside endoplasmic reticulum till telophase starts. All right. Now, next part of the question, why do centrioles duplicate? Now, as we have seen, the centriole has a role of releasing spindle fibers. And these fibers are going to pull the particular chromosome so that there will be a division of chromosome. All right. So, spindle fiber has a pulling ability due to which chromosomes are separated. Now, as we are talking about pulling ability, that means the pulling force should be acted from both the sides. If we are talking about tug of war, so in that tug of war game, is there only a single team? No, right? We have to create equal pressure on the both sides. So similarly, the centriole has to double. The centriole has to get double so that spindle fiber should be able to attach on both the sides of chromosome, on both the chromosome sides. Alright guys, so this was your logic behind this particular doubling of the centriole and what is happening of the parts of nuclear membrane. Alright, so let's move on to the structure of chromosome. Now after the condensation and coiling, what is the particular structure is going to look like? So the head of the chromosome is called as telomere. These are nothing but ends of chromosome. Now the middle part is called as centromere. This is your middle part of chromosome. Then the one arm is called as chromatid. So this is nothing but your one arm of the chromosome. If we are talking about the arms present on the same side, the arms present on same side, those are called as sister chromatids. Those are called as sister chromatids. And if we are talking about centromere, the attachment of spindle fiber takes place on a protein which is surrounding this particular centromere. This protein is called as here kinetochore apparatus or kinetochore protein. So this is nothing but here kinetochore protein helps for attachment of spindle fiber. So it is hook like a structure. It is hook like structure where your spindle fibers are going to get attached so that the pulling force will be applied from both the ends. Alright. Now there is going to be a short arm. This short arm is called as P arm of the chromosome, while long arm is called as Q arm of the chromosome. Alright, this is your basic chromosome structure which is required for this particular chapter. So let's move to your next phase, that is nothing but metaphase. So in metaphase, the complete disintegration of nuclear membrane has occurred and the centrioles are reached to their respective pole. As you can see here, the centrioles are reached to their respective poles. So these are nothing but my centrioles. Nuclear membrane is not seen, so nuclear membrane and nucleolus is completely broken down. And disappeared. Alright. So there is breaking down of nuclear membrane and nucleolus, and it is completely disappeared as well. But the chromosomes which were present anywhere inside a cytoplasm, they starts to arrange on a metaphasic plate, or it is called as equatorial plate. So these chromosomes are basically arranged in a line. So chromosomes are arranged on equatorial plate. Alright, so these chromosomes are arranged in such a way that all the chromosomes will lie on an equator of that particular cell, if we are considering that cell as an earth. Alright, so let's see what is happening. So there is complete disintegration of nucleus membrane and nucleus. We have seen that. The centriole and centrosome reach their respective pole. We have seen that. Now the spindle fibers start to attach on the kinetochore assembly. I have told you that whenever the spindle fibers are coming out, these spindle fibers are going to get attached in a hook-like structure called as kinetochore. Alright. The structure is with 2 sister chromatid and 4 arms. We have seen that if I'm talking about structure, this is your one sister chromatid and this is another sister chromatid with 4 chromatid arms. So there are 2 sister chromatids which is equivalent to 4 chromatid arms. Alright. So next one is chromosomes are arranged on the metaphase plate, or it is called as equatorial plate. Any doubt in this particular point, guys? If there is any doubt, please write it down in the comment box. Let's start with your questions. During cell division, spindle fibers attach to chromosome at a region called as kinetochore. We have seen that it is a hook-like structure on centromere of chromosome. Alright. Now, why do chromosomes have to arrange on equatorial plate? That is one of the interesting questions, guys. Why do they arrange? Because they have to experience the same force from both the ends. As we have seen, the centriole are going to release spindle fibers, and these fibers are creating a pressure and force on that particular chromosome so that these chromosomes will separate, and those is basically important for the division of nucleus. So in this case, the chromosome has to arrange on equatorial plate so that the equal force so that the equal force from both the poles and spindle fiber has to occur. If not, the distribution of chromosome becomes unequal. The distribution of chromosome become unequal, or there will be a damage on chromosome. That is non-repairable due to extra force. So that's why for equal force to generate on the particular chromosome, they have to arrange on a equatorial plate. All right. Now, what are the types of fibers? So there are total 3 types of fiber: polar fibers, kinetofibers, and astral rays or astral fibers. So now, what do you mean by polar fibers? The spindle fibers attaching from pole to pole. Suppose if this is my cell and this is your centriole, so there are spindle fiber which are attaching from pole to pole. This pole to pole attached spindle fibers are called as polar fibers. Alright. The spindle fibers which are attached to kinetochore. That means nothing but your chromosome attachment. That is called as kinetochore fibers, and these are one of the important fibers which are required for the separation of chromosome. So these are required for separation of chromosome. Now, as we have seen, the centriole is going to distribute the fibers in every direction, right? Some fiber will reach till chromosome, some fiber will reach till poles, but some fibers are moving backward. The fibers which moves backward towards cell membrane, those fibers are called as astral rays or astral fiber. Got my point, guys? These are the types of fibers. Now, the spindle fiber attached to kinetochore of chromosome, there is still an option of centromere given, but the fibers are attached to kinetochore, not centromere. Why is this happening? Because there should be equal division of centromere. Not centromere, as to divide the centromere equally. Alright. Next case is your anaphase. Right. Before that, I want to give you one question, that is, in the metaphase, whenever scientists have to study, whenever scientists have to study which phase is important for study of chromosome as its structure and number, as structure and number. So for this particular answer is nothing but my metaphase, Now why metaphase? Because after condensation of the chromatin fiber, all the chromosomes are arranged on the equatorial plate as a queue of students. Alright, and it is easy to hold all those on equatorial plate, and for number of chromosome, one technique is used called as karyotyping. Which is called as your karyotyping. Now, what do you mean by karyotyping? That means arranging the chromosome according to their length. So there will be arrangement of chromosomes according to their length. So that is nothing but your karyotyping, and that is very, very important for finding the number of chromosomes, and metaphase for determining or studying the chromosome as their structure. Alright. So now, after the metaphase is completed, the anaphase starts. As the spindle fibers are attached to the kinetochore, those are going to create a pulling force. Alright, and this pulling force is basically because of depolymerization. So there will be here depolymerization. So there will be depolymerization of spindle fiber. If you are pulling something, you are basically shortening that particular length. Alright. As the length is increased, your pulling force will decrease. As to increase the pulling force, we have to shorten that particular length of a rope. Alright, and the length of rope is shortened because of depolymerization. This will create more pulling force, and then the chromosomes are basically separated and moved to the respective poles. Easy. That is nothing but a case of anaphase. It is easiest phase of the complete cell cycle. Now, anaphase promoting complex is a protein degradation machinery necessary for proper mitosis of animal cell. Now, whenever we have to promote a cell division, we have to separate chromosomes. Alright, and these chromosomes are attached to each other. Whenever the anaphase promoting complex protein is activated, this means it will degrade the binding complex of protein between the chromosomes. So there is some complex which is bounding all the chromosomes together. Alright, and this complex has to break down whenever the anaphase has to promote. Alright, if APC is defective in human cell, that means the chromosome will remain attached to each other. The chromosome arms will remain attached to each other, and sister chromatid won't be able to get separated. All right. So here chromosomes, sister chromatids cannot be separated. Cannot be separated. So that means your chromosome will not segregate. Alright. So option number 3 will be my correct option. If you use a logic for Interphase, then and then only you will get this particular question on as per your 12th syllabus is concerned. Now what is happening inside the Interphase? That means chromosomes from the equatorial plate starts to pull apart towards the pole. As we have seen that the pulling force is generated, and this pulling force is basically pulling all the chromosomes toward the pole. The spindle fiber starts to depolymerize, as we have seen. To generate extra pulling force, we have to depolymerize that particular spindle fiber. The force is approximately 1 piconewton. This is extra information, guys. If you want to remember this, you can. Otherwise, you can skip this particular information. The tension is created on the fibers that causes centromere to separate. So, in the anaphase of mitosis, centromere separates. This is one of the important points. Alright. So let's move on to your question. Why are chromosomes pulled towards pole? This is basically a simple question. Now why towards pole? Because your centrioles are present over there. So the chromosomes are pulled towards pole as centrioles are present at that point, and there is depolymerization of spindle fiber, of spindle fiber. That's why your chromosomes are pulled towards the poles itself. Alright, easy peasy question. Now, can the force cause damage to the chromosome? That is one of the interesting cases. Chromosome will not get damaged, but the DNA present on the chromosome can get damaged because of the 1 piconewton force. So, based on experimental data, even I have worked on this particular case, that is 1 piconewton force is enough to break the DNA. When I was chosen to be NIG intern at National Institute of Genetics, Japan, so in that particular experiment and that particular research work, I was working with this particular case, that is this particular force is enough to break the DNA molecule. Alright, but this DNA molecule repair itself by their self-assembly manner. So 1 piconewton force is enough to break the DNA, but not chromosome. You can see damaged parts on chromosome, but they are of DNA. Alright, but this broken DNA self-repairs through intrinsic assembly. So the assembly of protein which is present inside the cell is very, very important, so that this particular broken DNA should be repaired and no mutations or the damaged DNA should be transferred inside the next cell. That is very, very interesting, guys. If you really want to get insights of that, you can go and check my work of research on the NIG intern websites as well. Alright. So the final phase of karyokinesis is called as telophase. If we are talking about telophase, in that case, your chromosomes are reached to the respective pole. First of all, your chromosomes are towards poles, As soon as they reach to your pole, this sends a signal. To whom? To ER. As the ER was keeping all our content safe. Those are nothing but your nuclear membrane contents. So this ER will release the content of nuclear membrane and nucleolus so that this will start to form a membrane around the separated chromosome. So this forms membrane around separated chromosomes. As you can see the dotted line, this is nothing but your nuclear membrane, and finally your nucleus is separated. The separation of nucleus is nothing but your separation of chromosome, so that after the chromosomes are separated, you can just have to wrap that particular chromosome inside the nuclear membrane. Alright guys, so nuclear separation process will be done inside the telophase. Now what is happening? Let's see. The chromosomes are separated towards the poles. Chromosome reach towards the respective poles. The centriole starts to move their respective position. Now the centriole which are present on the poles, they starts to move to their respective position, that is on the equator. Alright. Now the chromosome starts to unwind and elongate to become chromatin fibers. The chromosome can't stay as a chromosome inside the particular nucleus, and it can acquire the larger space. And we want to fit much of the biomolecules inside the smaller space. That's why this chromosome has to unwind and elongate, so that the exactly opposite thing that was taking place inside the prophase, it is happening inside the telophase. Alright, so telophase is not as such mind-boggling thing, but it is just a reverse of prophase. Alright, so in this case, your chromosome starts to unwind and elongate to become chromatin fibers again. So these fibers will start floating inside your nucleoplasm. Now the nuclear membrane and nucleolus starts to reappear. As we have seen, there is a dotted structure which is taking place. So your nuclear membrane and nucleolus starts to reappear, and spindle fiber break down and absorbed inside the cytoplasm. Now all the spindle fiber which are present are absorbed as their monomer. Alright, absorbed in a cytoplasm as monomers, which are again loaded with ATP molecule and used for further cell division if required. Alright, and two daughter nuclei are formed. So there is a formation of two nuclei. Alright, so let's see one question. Which of the following option gives correct sequence of the events during mitosis? First of all, condensation, right thing. Then telophase should be my last thing. Then your nuclear membrane disassembly. So these options are over. So there is nuclear membrane disassembly. Then arrangement at equator. So option 2 will be my correct option. So in the first phase, that is prophase, there is going to be condensation and nuclear membrane disassembly. Then finally at metaphase, there is going to be your equator arrangement. Then at anaphase, there is centromere division and segregation. And finally in telophase, there is formation of two nuclei is taking place. Alright, which of the following is not characteristic feature during mitosis in somatic cell? Alright, we have seen disappearance of nucleus. So it is a characteristics. Chromosome movement, during anaphase. So there is disappearance in prophase. Then there is movement in anaphase. And there is spindle fiber in metaphase and anaphase. metaphase and anaphase. So this is not occurring during your mitosis, but this takes place. The synapses occurs in meiosis. In meiosis, we will be seeing that in some time. All right. So next part of the mitosis to complete the particular cell division is cytokinesis. Now in cytokinesis, if we are talking about two cells, that is animal cell and plant cell, the things are slightly different. In animal cell, there is a formation of furrow. So there is furrow formation. Furrow formation. And there is microfilament ring, as you can see this. These are also called as contractile rings. These are also called as contractile rings. And these rings are basically moving from outward to inward. These rings are basically moving from outward to inward as a pinch of this particular cytoplasm is taken off. So it is basically pinching off cytoplasm where rings are moving from out to in. Alright, so in out to inward movement, the contractile rings are arranged in such a way that the cytoplasm is pinched off, and there is a deep groove has been formed called as furrow. After the formation of furrow, at one point, the breaking of this particular cytoplasm takes place and these two daughter cells are basically obtained. Two daughter cells are going to be obtained. Alright. While in case of plant cell, the thing is slightly different. That is, there is a formation of cell plate. There is a formation of cell plate. So all the vacuoles, the contractile vacuoles are arranged from in to out. Alright. So there is contractile vacuoles which are arranged from in to out in case of plant cell. Why they can't be pinched off? Because the plant cells having cell wall, it is tough to pinch off that particular cytoplasm. That's why from inside they have to generate extra pressure on that particular cell wall, and then only the particular cell is going to get divided. All right. This cell plate is further converted into your middle lamella, which is separating two plant cells. Alright, so this is nothing but my middle lamella, and this middle lamella helps to separate and connect plant cells, and that connection is known as plasmodesmata. I have covered that in the eukaryotic cell part of the cell, the unit of life chapter. You can go and watch that particular video for the extra information about plasmodesmata. Alright. So let's see what is cytokinesis is happening. That is, in case of animal cell, there is furrow formation. Contractile ring moves inward from outward, and these are nothing but made up of microfilament. These are nothing but made up of microfilament, which is one of the type of your cytoskeleton molecule. Alright. And in case of plant cell, there is going to be a contractile vacuole and ring moving outwards, so from in to out, and the cell plate formation is taking place. Animal cell can be easily pinched off because of lack of cell wall. Alright, there is only presence of cell membrane, due to which animal cell can easily be separated. So this was your cytokinesis, and congratulations, from one cell we have came up to two different cells. Alright, so let's see now what are the different questions. A cell at telophase stage observed by student. A cell at telophase stage observed by student in plant brought from a field. He tells his teacher that this cell is not like other cell at telophase stage. There is no formation of cell plate. There is no formation of cell plate. This is one of the important thing. And does the cell contain more number of chromosomes? That means the cell is unable to separate cytoplasm. All right. Now, due to which the total number of chromosome should be increased. All right. Whenever the total number of chromosomes are increased, that means the sets of chromosome are increased. The sets of chromosomes is basically increased, and whenever the set of chromosome is increased, that is called as your polyploidy condition. So option 1 will be my correct option for this particular question. All right. Let's see some of the video part, but the video is not running, guys. But all right, okay, yeah, it ran. So as we can see, the centriole move to your poles, then they start releasing spindle fiber. There is attachment to the chromosomes on kinetochore, and then the separation is taking place by pulling effect. All right. So in this case, your chromosomes are separated and this cytokinesis occurred. Due to this cytokinesis, your cells are basically separated. You could have seen every sequential event I have explained before in this particular video. All right. So let's move on to the significance of mitosis. Now, what is significance? That means there is only a formation of clones. That means identical daughter cells are basically formed, which are identical to their parent as well. Then any cell can perform mitosis. Yeah, there is no ploidy restriction. No ploidy restriction for mitosis to occur. When your cytoplasm to nucleus ratio is maintained, as it is well organized process, your cytoplasm is divided equally. Cytoplasm and nucleus is divided equally. All right. Then your chromosome numbers are maintained. Yeah, the numbers of chromosomes are same in the daughter cells and as well as in your parent cell. If the parent cell contain n number of chromosome, then even daughter cells are going to contain n number of chromosome itself. All right. The cells can be replaced with a new one if we are talking about wearing and tearing of the particular cell. All right. So worn out cells of the body can be replaced by the new ones because of mitosis. So in this process, the worn out cells are replaced. All right. And there is equal quantitative and qualitative distribution. Now, what does that mean? That means whenever I am dividing the cytoplasm, yeah, quantity is maintained. But what about quality? That means the protein distribution is also, the protein and ionic distribution is also equal. Due to which we can call it as your clones. All right. This was your significance of mitosis. But what are the disadvantages? Everyone talks about the advantages, but no one talks about the cons. So what are the cons for this particular process? That means only clones are formed. That is one of the cons. Even though the number of cells can get higher, and identical cell can get higher, but they are always remain identical. So they are always identical. So if one has some problem, other has same problem too. All right. So there is no genetic variation, as there is no recombination. So there is no recombination occurring. So that's why there is no genetic variations. They are not useful for the evolution, and some unwanted gene might get inherited. If there are some problem inside the gene, then that gene cannot be replaced as there is no genetic variation, no recombination, and that gene will be inherited in next generation as it is. Alright, these are some of the disadvantages of the mitosis. Now let's talk about something related to cell death. Now, what is cell death? Whenever the cell is going to die, it is either dying with a proper program manner process or it is dying suddenly. Alright, so if I'm talking about properly programmed cell death, that is called as apoptosis. In case of apoptosis, only single cell is dead. Only single cell is dead, and it is planned and programmed. Oh my god. It is like a serial killer. It is very much planned and programmed cell death. It's interesting, right? So in this particular case, your caspase proteins are involved. There are much caspase protein are involved, and they are in a cascade manner. What do you mean by cascade? That means like a domino. Whenever I am arranging any protein, they are in a sequential manner. So these proteins are arranged sequentially in the case of apoptosis. So there is surrounding cells are not affected, as only single cell is dead. That means surrounding cells are not affected. It is planned death and called as cellular suicide. Alright, the cell has to undergo suicide inside the phagocytic cell. So in this case, the planned program cell death, phagocytic cell will digest it. Phagocytosis by phagocytic cell, cells is going to take place. Alright. But while if I'm talking about necrosis, in that case, the damage to one cell kills the neighboring cells. Now that is interesting, guys. It is like a suicide bombing. It is like your suicidal bombing. Now what in this particular case is happening is that if there is a cell or there is a group of cell, and suppose if I am damaging the particular middle cell, then all the content are released out, and this released out content are basically damaging other cells. So it can be easily seen on the peel of banana. You can see the black spots on the banana, right? So this black spots is nothing but a process of necrosis which is taking place inside your case of cell death. Alright. So let's move to your next process of cell division, and this will be our final topic of this particular chapter. Alright. That is nothing but meiosis. Now, meiosis is generally called as reductional division. Now, why is this particular reductional division name came from? So, in case of meiosis, the number of chromosomes in daughter cells are half that of parent cell. That's why your meiosis is basically called as reductional division. Now, in this particular case, as soon as I am halving this particular chromosome number, that means I should go with a cell having even number of ploidy. Alright, so the cell should be having a ploidy of diploid, tetraploid, hexaploid, octaploid, etc. So there should be even number of ploidy has to be there if I'm talking about meiosis. So the cells with only even ploidy can enter in this particular process. The process is not used to cover up worn out cells. Now generally whenever I'm talking about meiosis, as the chromosome numbers are going to get half, that means they are not leading to the same cell again. Alright, so there is no similar cell is forming. That's why there is a formation of gametes, and this gametic fusion is then going to lead to the same ploidy condition again. Alright, The reduction of the number is covered up whenever the gametes are going to get fused. The germ cell of a body are formed, that is nothing but sperm and ovum, which are having haploidy, that is single set of chromosome, and the process is used to form gametes, as I have told you already. Alright. So let's see what is meiosis is comprised of. If I'm talking about meiosis, the meiosis is comprised of two phases, that is meiosis 1 and meiosis 2. Now the meiosis 1 is reductional phase. Now the meiosis 1 is a reductional phase. That means the total number of chromosomes are going to get half in the process of meiosis 1. That means if there are 2n number of chromosomes, after meiosis 1, I am going to get n number of chromosome cells, and finally these are divided into 4 cells. So in meiosis, we have to remember that one cell is going to give me 4 different cells. Like in mitosis, it was 2. In case of meiosis, it is going to be your 4 number of cells. Alright. Then there is going to be the meiosis 2. The meiosis 2 is your equational division. Just similar to the mitosis, but the recombinants are going to be same. It is similar to mitosis, just recombinants are going to be formed. Alright. This is nothing but a case of meiosis 2. So as meiosis 1 suggests, the interphase occurs before meiosis 1, and throughout the process, the interphase is not repeating again. Alright. So in this particular phase, your cell is undergoing interphase, and after the interphase is completed, the M phase is going to start. So here M1, and finally there will be M2 phase is going to take place. Now in case of meiosis 1, the process is reductional, and karyokinesis and cytokinesis take place as usual in every cell division. While if I am talking about meiosis 2, it is equational division, and both karyokinesis and cytokinesis is going to take place. Alright, easy peasy. So if I am talking about meiosis, in the case of meiosis, how this particular system is going to work. Suppose in G1 phase, the cell has N content of DNA, N content of DNA, sorry, 2N content of DNA. So in S phase, as we were seeing that there is even ploidy cells are entering, there will be 4N content of DNA. 4N content of DNA, and we have to divide this particular content equally within every cell. So in case of G2 phase, there will be division. There will be not division. There is the content remains same, that is 4N content. So in G2 phase, the content of DNA is 4N again, but as entering inside M phase, after the completion, there is formation of 4 cell with N content, N content, N content, and finally your N content. So there are total 4 different cells are formed after your single cell undergoing the meiosis with a ploidy of even set of chromosomes. Alright. So what is this particular meiosis will look like? Now the meiosis 1 is basically comprising of your interphase, prophase 1, pro-metaphase, that is nothing but your just before metaphase, that is metaphase 1, anaphase 1, we have to skip this particular part, metaphase 1, anaphase 1, and telophase 1. Then cytokinesis take place, and finally your prophase 2, metaphase 2, anaphase 2, and telophase 2 is going to occur. Alright. Now the interesting part in the meiosis is basically your prophase 1, from which your questions are built in the NEET. Alright. So meiosis has evolutionary significance because it results in recombination, guys. So meiosis is basically helpful for recombinations. Alright. Now how does this question can be solved if we are not aware about recombination within meiosis. All right. Evolutionary significance. All right. Whenever I'm talking about evolutionary significance, that means the evolution has to move in a positive manner. In a positive manner, that means new species has to form. Whenever you want to get a new color, what do? You just recombine all the colors you have, then you are going to get some new color. All right. So similarly, in this particular case of meiosis, you are going to see some recombinations, and these recombinations are helpful for the evolutionary significance. All right. Let's move to your meiosis 1 with a karyokinesis 1. Now, the karyokinesis 1 is having its first phase, that is prophase 1. And this prophase comprises of 5 different stages. So there are 5 stages in case of prophase 1 of the meiosis 2. Alright. The first phase is, before that we will be moving to your question. In the meiosis, crossing over is initiated at your Pachitin stage. Alright. We will see that what is going to happen. Alright. So first of all, leptotin is the first phase of prophase 1. Now, if I'm talking about leptotin, in leptotin, obviously, as it is your first phase of prophase 1, that is early prophase. That is early prophase. And in case of early prophase, we are going to see chromatin fibers. Alright, they will start undergoing condensation and coiling. We have seen that in the case of mitosis, and all chromatin fibers are present in bucket stage. This is very, very important as per your match the following is concerned. So this chromatin fibers are present in bucket stage. If the NEET aspirants are preparing for this particular chapter, they should be known about the typical terms related to every phase of this prophase 1. Alright, this is your primitive phase where nuclear membrane is still intact, just there is a coiling and condensation of chromatin fiber is taking place. Alright, moving to your next phase, that is zygotin. So in case of zygotin, the chromosomes are formed, but these chromosomes are basically having your pair of each other. Alright, so they are going to find their own pair and they are matching them through their homology. That means complete similarity. So the structure and function of pair of chromosome is same. Chromosome is same, and such chromosomes are called as homologous chromosome. All right. The chromosomes start to look around to find homologous chromosome. Where is my companion? I can't see that companion in the complete nucleoplasm. That is the question the chromosome is going to ask. And these chromosomes are basically attached to each other. Alright. And after the pair formation, there is a synaptonemal complex. So the protein complex is going to take place. This protein complex helps to join this particular chromosome together. And there is going to be the synaptonemal complex. Alright. This synaptonemal complex helps this chromosome to remain attached to each other. Alright. They are obviously tied up with the partner, and these partners are remain attached in the process of zygotin, and that process is called as synapsis. Alright. So the synapsis is formation of synaptonemal complex, so that homologous pair of chromosome remain attached. Chromosome remain attached to each other. Alright, this is the process of zygotin. So in case of leptotin, we saw bucket stage, and in case of zygotin, we saw synapsis. Next case is Pachitin. As the companions are attached to each other, they try to mingle with themselves. Alright, and during that mingle, they just overlap their chromatid arms on each other. You can see that here. that the chromatid arms are overlapped on each other, and as soon as the overlapping is taking place, that is called as crossing over. The point or the part or the region on which the crossing over is taking place is called as chiasmata. So for patchitin, there is crossing over word is important, and chiasmata formation is very important. You can call this as chiasma as well. Alright. The chromatid arms of the chromosomes cross over each other. Alright. They start overlapping, and this overlapping forms a chiasmata. Now, the patchitin is very important, guys. If any NEET aspirant is preparing for NEET, then your patchitin phase has to be very clear about the meiosis. In case of patchitin, there is formation of bivalent. Alright, there is a formation of bivalent. Now, what do you mean by bivalent? That is, there are two centromeres. There are two centromeres, but there are total 8 chromatids. Alright, right? So 1, 2, 3, 4, 5, 6, 7, 8. So there are 2 centromere. There are 2 centromeres, but there is 8 chromatid arms. 8 chromatid arms. That means there are total 4 sister chromatid. 4 sister chromatid. You should be very good with this particular convergence. Alright. Now the process is helpful. As soon as the overlapping is taking place, that means the character from one chromosomes are entering into the another. Right? So there is genetic exchange. There is genetic exchange. The genetic material from one chromosome is transferred to another chromosome in the total patchitin phase. So there is going to be the process of recombination taking place. And in this process, the enzyme recombinase enzyme is going to play a crucial role. Alright. So let's move. What is bivalent? We just saw that there are total 2 centromere and 4 chromatids. Alright. So here we are talking about chromatids, but in the answer we are talking about sister chromatid. Alright. So they are not talking about the separate chromatid arms, but they are talking about the sister chromatid arms. Alright. So there are 4 chromatid and 2 centromere. Alright. Now, the next stage is diploting. Alright. After crossing over, as soon as the hands are shaken between your chromatid arms, then they think that why should we have this particular synapses bond? Why should we remain attached to each other completely if we are holding our hand together? Right? So in this particular phase, your synaptonemal complex starts to break down. So here we have mentioned that synaptonemal complex breaks down, and it is a reverse process of the synapses. Whenever any reverse process is taking place, we use the word D. And which process is that reverse? That is synapses. So there is desynapsis taking place in case of diploting phase. Alright. And the final phase, which is also one of the important phase, where nuclear membrane starts to fragment, or nuclear membrane and nucleolus starts disappearing. The centriole centrosome duplicates and starts to move their respective pole. That is similar to your late prophase. Alright. So this is common as your late prophase. And here the chiasmata, which has been formed by overlapping of the chromatid arm, will try to shift to the terminals. Now, why is that happening? As soon as the synaptonemal complex is broken down, as soon as the synaptonemal complex is broken down, that particular point, the chiasmata start to shift. Alright, and they try to hold themselves with the fingers. Alright, so the chiasmata is shifted towards terminal end of chromatid, and that process is called as terminalization. So we have came across different terms If we are talking about leptotin, that is bucket stage. If we are talking about zygotin, there is going to be synapses. If we are talking about pachytin, there is going to be a crossing over, recombination, or chiasmata formation. If we are talking about diplotin, then there is desynapsis, that is the reverse of synapsis. And finally, in case of dikinesis, there is going to be a terminalization. So this homologous pair chromosomes are basically formed, and now they are exposed towards your spindle fibers. So let's see what is the question. Arrange the following events of meiosis in a correct sequence. Now, as we know that disappearance of nucleus is taking place inside dikinesis. It was the last phase, right? So option 1 and 4 are wrong. Now terminalization of chiasmata, this is again in dikinesis, alright, but that is taking place before the disappearance of nucleus, alright. So 3 will come before 4, 3 will come before 4, alright. Synapsis and crossing over, this was taking place inside zygotin, and crossing over was taking place inside pachytin phase. Alright, so the sequence was leptotin, zygotin, pachytin. Alright, so first of all 2, then 1, 3 and 4. So option second is my correct option for this particular sequence. Next question, during gamete formation, the enzyme recombinase participates in. Now as we know, the recombinase, is helpful for recombination. Alright, so it is very helpful for recombination, and this recombination was taking place inside pachytin. It was a part of prophase 1. It was a part of prophase 1, and this prophase 1 was a part of meiosis 1. Right? So that's why your prophase 1 will be the correct option. That is option number 3. Now what is metaphase 1, guys? Now from this onward, the chapter becomes very easy, very easy. Alright, if you are very well clear with the mitosis, then this particular chapter won't have any problem from this particular point. If I am talking about metaphase 1, in this metaphase 1, the nuclear membrane and nucleus disappears completely. That means your pair of homologous chromosome, is exposed. Where does it exposed? It is exposing towards the spindle fibers. Alright. So now the centrioles are reached to their respective poles. Now this will start releasing spindle fibers. So start releasing spindle fiber. Now what does happen in the metaphase? Yeah. All chromosomes, that is the pair of homologous chromosome, now will arrange on the equatorial plate, and the spindle fiber starts to attach on the kinetochore assembly. That is exactly similar to the metaphase of the mitosis. Alright, let's move on to the anaphase. That is, anaphase is again pulling of this particular chromosome. But will that separate the centromere? No. Why? Because in case of mitosis, there was a single centromere present. But if I am talking about meiosis 1, there are 2 centromere and 4 sister chromatid. Right? So there are 2 centromere and 4 sister chromatid. That means your centromere won't be separate. Alright? Centromere is separated completely independently. Alright? So there won't be, if suppose this is my centromere, this is not going to divide in case of anaphase 1. So there is no division of centromere. That is the only trick, or the tricky question can be asked inside your anaphase. Based on assertion and reason type question or statement wise question in the NEET, this is one of the hot topic, or we can say this is one of the important topic we should know about if I'm talking about anaphase 1. The chromosome with the part of homologous chromosome on the chromatid, that means if this is a complete red chromosome, there is a part of blue chromosome as well. This has been taking place because of the recombination process. Alright. So now the chromosome with the part of its homologous chromosome is pulled towards the pole. Now there is again same thing, that is nothing but depolymerization of spindle fibers is going to take place, but centromere is not separated in this particular phase. This is one of the important things. Only homologous chromosomes are separated independently. Alright. Now it creates a pulling force on the chromosome, and similarly guys, there should be a APC, that is anaphase promoting complex, should be activated. Then and then only the anaphase will be promoted. Alright. Finally, the telophase, as soon as your chromosome are reached to the respective poles, there will be unbound and elongation of the chromosome and converted into chromatin fibers. There will be disintegration of nuclear membrane and nucleolus, which will be start to reform. That means this telophase 1 will send a signal to ER, endoplasmic reticulum, and there will be reappearance. There will be reappearance of nuclear membrane, and nucleolus Alright, easy peasy. And the two nuclei are formed with the haploid set of chromosome. Alright. So in this particular phase, as we have seen that meiosis 1 is a reductional division. So if I am taking a cell starting with 2 N ploidy, after end of meiosis, I am going to get 2 cells, but that with the haploid set of chromosome. Alright, this is one of the important case for meiosis 1 only guys, for meiosis 1. Alright. So let's move to your cytokinesis. As soon as I have divided my nucleus, then there is only the process which is taking place, that is cytokinesis. That is nothing but division of cytoplasm. Now it is again similar. Division of cytoplasm is going to be similar, exactly similar to that of the mitosis. Cytokinesis don't change in either mitosis or meiosis. So cytokinesis is not changed according to type of cell division. According to type of cell division. Alright. So if I'm talking about animal cell, then there is again the same thing, that is furrow formation. And if I'm talking about plant cell, there is going to be the cell plate formation taking place. Now, after the meiosis 1 is covered, there is no interphase taking place. So meiosis 2 starts immediately after cytokinesis 1, and it doesn't halt for interphase. So there is only a single interphase stage running guys in the case of meiosis. Alright, even though the stages or the processes are two, that is meiosis 1 and meiosis 2, for the complete process the interphase is run only once. Alright, if I'm talking about meiosis 2, there is going to be the prophase 2. Alright, similar to your mitosis. So this is exactly similar to mitosis. Alright, one chromosome with two sister chromatin. So there is one centromere and there are two sister chromatids. Alright, so there is one centromere, two sister chromatid. Then this complete chromosomes are basically arranged on the equatorial plate in case of metaphase 2. The centrosome and centriole move to their respective pole. They start releasing spindle fiber. There will be a pulling force inside anaphase 2. The chromosomes will be separated, and finally inside telophase and cytokinesis I am going to get 1, 2, 3, 4 cells are going to be obtained. Alright, the two cells which are obtained the two cells which are obtained after meiosis 1 are entering inside the meiosis 2 together. So basically, two cells obtained after meiosis 1, two cells obtained after meiosis 1 are entering and processing the phase of meiosis 2, the phase of meiosis 2 simultaneously. So there is no subdivision of prophase 2 of meiosis 2. All right, as there was some or 5 subphases of prophase 1 in case of meiosis 1, there are not as such any phases in case of meiosis 2. All right, it is just similar to your mitosis. The difference is you are going to get recombinant at the all right. Anaphase 2, Anaphase 1 are one of the important phases whenever we are talking about aneuploidy or polyploidy condition. All right, guys. So that APC you should be remember properly, because if we are talking about genetics in some other videos, then in that genetic topic we will be needing this particular concept very badly. So I am covering up this particular topics which are fundamental of the biology and very, very important as per your NEET point of view is also concerned. All right. So let's move on to the significances of the meiosis. That is, they are helpful in the gamete formation. In gamete formation, the recombinant gametes can lead to variation. All right, there will be inheritance of information. That means from a male parent and female parent, I am going to get some new information together inside the particular organism. So this inheritance is taking place from both the parents. So meiosis is basically involved whenever there are two parents for the formation of individual are required. Then due to the patchotin phase, there is genetic variation. So there is going to be recombination in patchotin. Phase, where you are going to see the genetic variation taking place. Along with that, the particular chromosome of the parent is also going to decide the genetic variations. So these genetic variations are also decided by type of genes on parental chromosome. All right. Then this genetic variation is promoting evolution. Obviously, as the survival of fittest is the concept of Darwin, whoever will have the better genes will survive, and that will promote evolution further. And there are more number of cells are going to be formed, as one cell was going to give me 4 different cells. All right. So there is more number of cells are basically formed. So as these were pros, we were also talking about the cons. So what are the cons of meiosis? That is nothing but only even ploidy cell can enter in this particular process. There is ploidy restriction. So there is a ploidy restriction in case of meiosis. All right. The fusion of good gametes is a chance factor. If we are talking about the separation of genes, and if we are talking about some bad genes overlapping in the case of recombination, this bad genes can also get inherit in the next generation. That is completely a chance factor. All right. So the gametes can have good or bad genes. Based on the recombination, guys. Based on recombination. As the recombination was helpful, this is also some disadvantage to the meiosis. Now the chromosome number gets halved. So in this particular process, whenever we are halving the particular chromosome number, that means I'm increasing the number of gametes, and that will decrease the proper gamete to fuse to the new gamete, that is a female gamete or a male gamete. All right, and there is a chance of chromosome mutations are very, very high. So in case of recombinations, if we are talking about the bad genes, so the recombination can also lead to some of the mutations. So the mutations can take place, but this is not directly with the process of meiosis. All right. So I think we have covered all the topics of this particular chapter along with various questions, also PYQs of the NEET, and hopefully you will be learning this chapter very, very clearly. If you have any doubts, any questions, please DM us on the Instagram, and also mention your questions in the comment box so that I can give you a response properly for that particular questions. All right, guys. Thank you, guys. Please do not forget to share, like, and promote this video within your friends so that everyone will get new insights about the cell division and cell cycle, and everyone will be well prepared before the NEET. All right, guys. Bye bye. See you in the next video.